Good morning, student. We all are going through a constraint phase. So it's very difficult for me to teach you without seeing you, without interacting with you. But we have to adapt some new techniques. So I hope you will all bear with me. So let's start. Uh, Today our topic is concept of antigen. It's very new to you and not well covered in your book. So go through it properly and ask me if there is any type of queries or mail me back. Okay. So let's start. So in this slides and in this topic, after completion of this topic, we will able to know the definition of antigen, their chemical natures, their types, few definitions like hapten, superantigen, etc. The factors which determines the antigenicity, what is epitopes or antigenic determinants, and what are the uses of antigen and the detection method for the antigen. So first we begin with the definition. So what is antigen? This antigen is any substance when introduced into the body which can stimulate the production of antibody. So the word antigen originated from the word antibody generation. Anti from antibody, gen from generation. So the substance which is capable of introducing or capable of producing antibody when introduced into the body are called antigen. So what are immunogens? Immunogens are molecules which induce an immune response. Okay, so are they different? That is, antigen is different from immunogen? Yes, in some sense, because all the immunogens are antigens, because the immunogenicity or immune response can be produced by production of antibodies. So all the immunogens are antigen, but all the antigens are not immunogen because all antigen may produce antibody but cannot elicit the proper effective immune response by itself. Example is haptens. So we'll discuss about hapten later on. So remember that all immunogens are antigens, but all antigens may not be immunogen. Okay. Now, what are the chemical nature of antigen? The most potent and most common type of antigens are protein in nature. Proteins are mainly in the form of glycoprotein, lipoprotein or pure proteins. They are the most potent antigen, very good immunogen, and most of the antigens are protein in nature, but remember that all proteins are not antigenic in nature. The protein is followed by polysaccharides, pure polysaccharide as well as lipopolysaccharides. Both can be act as good immunogen. But nucleic acids, they are usually poorly immunogenic and they can produce or depict or elicit an immune response only if they present as nucleoprotein, and followed by lipid, and that lipid is generally non-immunogenic in nature, although they may act as hapten. We will discuss about hapten later on. So protein is good immunogenic followed by polysaccharide, whereas nucleic acid is poorly antigenic and lipids are non-antigenic. Now, what are the types of antigen? The antigen can be classified based on certain categories. Number one is based on their ability to carry immunological reaction. Number two, based on their origin. And number three, based on the immune response they produce. So according to the immunological reaction, they can be classified into complete antigen and hapten or incomplete antigen. According to the origin, they may be exogenous or endogenous, and according to the immune response, they may be T cell dependent or T cell independent. There are other some categories which are very important, like superantigen, histocompatibility antigen or transplantation antigens, and tumor antigens. They are special type of antigens we will discuss. So we'll go one by one.
Now the first one, that is based on ability to carry immunogenicity and immunological reaction. So according to this, it can be classified into complex antigen or complete antigen and partial antigen or hapten. So what are complete antigens or complex antigen? The complete antigen is able to induce antibody formation and produce a specific and observable reaction with the antibody produced. So mainly two properties. Number one, induce antibody formation, that means immunogenicity, that is production of antibody when the antigen introduced into the body. So the immunogenicity. And number two, produce a specific and observable reaction, that means immunological reaction. So not only production of antibody, that is immunogenicity, also production of a visible immunological reaction. So these are the two main properties of complete or complex antigen.
Then partial antigen or incomplete antigens, they are also called hapten. They are number one, mainly non-protein in nature. Number two, they are low molecular weight substance. And number three, that is they are incapable of inducing the immune response by itself. And number four, they can elicit immune response when coupled with a carrier protein. So, four special characteristics of partial antigen. They may be non-protein in nature. We have also seen in the example of lipid antigen, they are non-antigenic. Example, hapten, which may be antigenic in nature. So they are mainly non-protein in nature. They are low molecular weight. Number three, they cannot elicit immune response by themselves. But number four, they elicit the proper complete immune response when combined with a carrier protein. So what are the carrier proteins? Carrier proteins, they are non-antigenic component. They itself also non-antigenic component, but helps to provoke the immune response when combined with hapten. The example includes albumin, globulin, etc. So the hapten molecules and the carrier protein both are non-immunogenic in nature, but when the carrier protein is attached with the multiple haptens, then it forms a complete antigen which induce or elicit the immune response. So this hapten again can be classified into complex hapten and the simple hapten. So what are the difference? We all know about the valency. Here the valency means, in case of antigen, the valency means that is the combining site for the antibody. If there is only one site for combining with the antibody, it is monovalent antigen. If there are multiple sites, two or more than two sites in the antigen for combining with the antibody, they are called polyvalent. So complex hapten, generally they are polyvalent, so they have two or more antibody combining sites, whereas in simple hapten they are monovalent, so only monovalent or univalent, so only one antibody combining site. For precipitation reaction, we need multiple sites in the complementary sites in antigen and antibody, so they can form a large precipitate. So complex hapten can participate in the precipitation reaction, whereas simple hapten cannot. So hapten is important for your short answer type question, small question. So you will discuss the hapten is classified according, if the antigen will classified according to their nature of action or immunological action. The incomplete or partial antigens are called hapten. They have three characteristics, number 1, 2, 3 and 4. Then the example of carrier protein and the different types of hapten. So you will get the almost the full marks. Now, based on origin, we can classify the antigen into exogenous and endogenous. So what are exogenous? Exo mean outside, genus mean generation. So generated outside from our body. So they generated outside, so they must be introduced into the host body from the surroundings, from the external environment by inhalation, injection, any other methods. They are basically pollutants, microorganisms, pollens, drugs, etc. So from the outside they enters into the host body through endocytosis or phagocytosis. Then they present to the antigen presenting cell or APC and processed into fragments for generation of the immunogenic reaction. Whereas endogenous antigen, they generated within the cell. How? They generated within our body, within the cells of our body as a result of normal cellular metabolism or because of some viral or intracellular bacterial infection. So they generate within our body endogenous antigen through normal metabolism or viral or bacterial infection, and exogenous antigens, they introduced or they enters into our body through phagocytosis or by endocytosis, then present to the APC and processed and produce the immunological reaction. Now, according to the endogenous antigen, this endogenous antigen again can be classified into three main categories. Number one is allogenic or homologous antigen. Number two, xenogenic or heterologous antigen, and number 3 is autologous antigen. So number 1, that is allogenic or homologous. Homologous means homo means same. Okay, so why this is called allogenic or homologous? Because these type of antigen present in some individual of a species, whereas absent in other individuals. So they not present in not all individual of the same species, present in some individual of the same species according to their genetic preference. So present within a species, within a same species, that is why it is called homologous. But they are allogenic because they present in some individual and absent in certain individual according to their genetic preference. These type of antigen also called the isoantigen. And the example, very common example is blood group antigen. We have some of you have B plus, some of you have A plus, some of you have AB, some of us has O. So they are different in different individual of the same species. Okay. Then the xenogenic antigen or heterologous. Hetero means different. So why this is called xenogenic or heterologous? Because here the antigen is shared by the different species, even can be present in the plant also. So one capable of stimulating the production of antibodies that react with the tissues from other animals of the different species or even with the plants. So the antibody produced by these type of antigen can react with the animals of the different species or plants. So that is why it is called the heterologous, different species, heterologous antigen or xenogenic antigen. Now, autoantigen by autologous antigen. Why it is called autoantigen? We know that immunity is mainly the fighting against the foreign body which introduced into our within our system. So our own antigen usually not produce any visible immune reaction within us because they already exposed, already sensitized to our immune system during the birth process. So they generally not elicit the immune response. But sometime due to the altered genetic response or altered environmental factors, our body treat our own antigen as foreign substance and produce the immunological reaction. So our own antigen behaves like the foreign particles and autoimmune diseases result from these type of autoantigens. So an autoantigen is usually a normal protein or complex of proteins that is recognized by the immune system of the patients suffering from a specific autoimmune diseases. These antigens under normal condition should not be the target of the immune system, but mainly due to some genetic and environmental factors, their normal immunological tolerance has been altered or lost for these patients and produce some autoimmune diseases. Example includes nucleoproteins, nucleic acids, etc. Now, based on immune response, we can classify the antigen into T cell dependent antigen and T cell independent antigen. T cell dependent means the antigen requires the help of T cell for the production of antibody by the B cell. And whereas in independent case, the antigen directly or itself can stimulate the production of antibody. So, example of first category is erythrocytes or serum proteins, different serum proteins, and for the second one, it is pneumococcal capsular polysaccharides, flagellin, etc. Now, some special categories of antigen. One of these is super antigen. Why it is called super? When we call super? Because extraordinary, above normal, beyond normal. So super antigen. So normally one antigen can stimulate only maximum 1%, even 1 in 1000 T cells. But in case of some antigen, almost 25% of the T cell gets activated. So these super antigens are that type of antigens which polyclonally activate large fraction or number of T cells, up to 25% for a single response. So example being the staphylococcal enterotoxins, which cause food poisoning, streptococcal pyogenic exotoxin, and some mouse memory tumor virus. There are many. So super antigen is important for the short note. You should go through the definition example along with the picture. Remember to draw picture if there is any short note on super antigen. Okay. Now another special type of antigen is histocompatibility antigen or transplantation antigen. They are mainly related with the graft rejection. What is graft rejection? You may be new to this term. Graft rejection means when some organ or some skin flap or any tissue transferred from one donor to another recipient, then this type of organ or this donated organ or tissue section, this is called graft. Okay, so they must be matched in two individuals, and otherwise it will our body will treat it as foreign particle and reject it. So and if after transplantation it will be rejected by the recipient, so there is no good occur. That is why the compatibility, that is the compatibility for the graft transplantation is very necessary, and the antigen related with these type of reaction, transplantation reactions, are called the histocompatibility antigen or the transplantation antigen. Another special type of antigen is tumor antigens. It is very important for the medical ground because it is very helpful for diagnosis of the malignancy or malignant cell, because these tumor antigens present in excess amount or even exclusively present in the malignant cell or the carcinogenic cell, whereas it may be absent in the normal cell or may be present in very minimal quantity in the normal cell. So detection of these type of antigen in a particular person is obviously suggested or drag us to the diagnosis of particular malignancy. That is why tumor antigens are very important for the medical personnel. Some examples are alpha-beta protein, which normally present in the fetus but again reappear in some carcinoma like hepatoma. Another one is carcinoembryonic antigen, and there are many. Okay, so after that we will go for the factors which determine the antigenicity or immunogenicity. So they are called the determinants of antigenicity or immunogenicity. There are so many factors. We will discuss it one after another. First, the size. We need an optimal size to produce optimal response. Okay, just like enzyme action, optimal pH, optimal temperature, optimal concentration, just like that for production of immunological reaction also we need an optimal size of the molecular immunogen to produce the optimum response. Generally, the smaller molecules may either very weakly immunogenic or non-immunogenic. Just like that, hemocyanin, a large molecule, and they are very highly immunogenic in nature, whereas hapten আমরা আগে পড়লাম, we have studied earlier that haptens, they are low molecular weight and they are very less or non-immunogenic. Number 2, chemical structure we have already studied, that is proteins followed by polysaccharides are potent immunogen, whereas nucleic acid is poor immunogen and lipids are generally non-immunogenic in nature. Number 3, foreignness. We all know that the antigens are generally foreign to us. So antigens which are foreign to the individual, that is non-self, they induce an immune response, and normally our self antigen, as they already exposed to our immune system gets sensitized during the process of development. So our self antigen are generally non-immunogenic in nature, and wherever this breach of this mechanism or this system, they produce the autoimmune diseases. But some self antigen are not generally treated as foreign body. They like lens antigen, sperm antigen, etc.
Now, according to susceptibility to tissue enzyme, we can say that substance which can be metabolized in our body with the help of our own enzyme system, they are actually good antigen. The substance which cannot be metabolized or the enzyme are not present to metabolize the particular substance, these type of substance are generally not good antigen to us. Example being the D amino acid. We all know that our body composed of L type of amino acids, and we lack the enzyme which metabolize the D amino acids. So D amino acids are poorly immunogenic for human being. Same for polystyrene latex. Now, for the antigenic specificity, that is, it's depend upon the position of antigenic determinant group in the antigen molecule. We will discuss it later on when we discuss about epitope, that is the antigenic determinant. Now, for species specificity, that is, the antigen can be present in the all member of a same species, that is, this antigen is specific for this species. This type of antigen, that is, this species specificity has got certain very important use, because this species specificity actually prove the phylogenetic relationship, that is, we belong to same species, and that's why it can be used to trace the evolution, that is, from here come this, and from this come that. Okay, and it is very important for forensic science. How? That is, if we find some blood stain, if we find some hair, etc., then after analysis, this special species specific antigen can help to diagnose which species this the sample belongs to, which species. Is it for animal origin, it is for human origin, or like that. So it's very important for forensic application. And then comes the isospecificity. We have already discussed during the classification of antigen, endogenous and special type of endogenous antigen. So isospecificity is quick revision, that is, it is present in some but not all individual of a specific species. So it is species specific, present in the particular species but not in all individual of that particular species. So this presence and absence in the individual of the same species is determined genetically, just like example being the blood group antigen. Okay.
Now, autospecificity. Autospecificity, that means self-antigen are usually non-antigenic. We have already gone through this, but there are few exceptions like lens protein, sperm protein. They are, though they are self, but they are treated as foreign. Why? Because during the developmental stage, they are not exposed with the immune system. So immune system not sensitized to these lens protein or sperm protein. So whenever there is injury, these protein liberated in our system and our body treated them as the foreign particles. So, and develop the autospecificity or autoimmune diseases. Then the heterophile specificity. We have also we have some idea, already have some idea on heterophile specificity. That is, the antigen is related or present in the closely related group of same different species also. Okay, so shared by the different species, even the plant present in the closely related different species. Okay. Example, the antigen used in Well-Felix reaction, antigen used in Paul-Bunel reaction. You will go in detail in your microbiology section in second year, second prof. Then the next one is organ specificity. What is organ specificity? That is, some antigens are confined to particular organ, like some are present in the brain, some are present in the kidney, some are in the lens proteins, and so they are very organ specific, but they can be shared by in the different species. So they are present in the kidney of human being, kidney of another animal, kidney of another type of another species of animal. So they are organ specific but not species specific. So example include the brain specific antigen, which can be shared, which is shared by human as well as sheep. Okay. So these type of specificity, that is the species specificity, organ specificity, isospecificity, these are related with the autospecificity. These are related with the genetic factors, that is, some gene or genetic factor alter or regulate the response of this type of specific antigen. Okay.
Next comes the age. Now we are very accustomed with the fact that the more than 65 years and less than 10 years are not allowed to go outside even with mask. Why? Because in case of old individual and for the very young individual, they lack the ability, or they have diminished ability to produce proper immune response against certain immunogen. So actually the extreme of age group may be due to lack of proper maturity or due to senile degeneration. Their immune system are not as responsive as the young age group or adult age group. So this is the age specificity for the antigen. Now come the degradability. What is degradability? That is the antigen which can be easily phagocytosed or endocytosed by the body and present to the APC so that it can be degraded or fragmented further. These type of antigen are more potent than the antigen which cannot be phagocytosed or degrade easily. So as because the most of the antigens, that is the T-dependent antigens, for the proper development of immune response, it requires to be phagocytosed and processed and present to the APC and then fragmented and to elicit the response. This type of degradability is very important for proper depiction of the antigenicity.
Now come the dose of the antigen. An optimal dose of the antigen is very necessary for production of optimal response. Any dose above or below this optimal dose is not produced, will not produce the desired response or the desired immune reaction. Next one is route of administration. This route of administration is very important because one route may be better than other, and it also depends on the route of administration. The nature of response also can be altered. So we can see that subcutaneous route is better than the intravenous or intragastric route, and the route of administration also altered the nature by that this way, that is antigen administered intravenously will carry it first to the spleen, whereas the antigen administered subcutaneously first moves to the local lymph nodes. So the nature of response also varies according to the route of administration. So after discussing the factors which determine the antigenicity, we will go for the adjuvant. Adjuvant is also very important term. What are adjuvants? Adjuvants are the substance that when mixed with an antigen and injected with it boost the immunogenicity of the antigen. So remember that adjuvant is not carrier protein. Carrier protein, when hapten along with carrier protein, they produce the complete antigen or produce immunological reaction. Hapten itself cannot produce immunological reaction. Carrier protein itself also non-immunogenic. But adjuvant is the substance which when mixed with antigen it itself can produce immune response. Okay, but after addition with the adjuvant, their immunogenicity increased or boost. The adjuvant can boost the immunogenicity of the antigen when mixed with an antigen and injected. So some examples, few examples are aluminum hydroxide, aluminum potassium sulfate or alum, etc. These adjuvants has got some advantage and some disadvantage. So what are the advantage? These adjuvants are used for with the vaccine. Okay, so what are the advantage? First, it boosts the immune response. So it is very important when the antigen has got low immunogenicity, and when very minimal quantity of antigen is available. So when the quantity of the antigen is suboptimal or the immunogenicity produced by the antigen is low, then the adjuvants are very important because they boost the immune response. Number 2, how they boost? Because they enhance the co-stimulatory signals by prolonging the retention of immunogen also. So they enhance or boost the immunogenicity by two mechanisms, that is it enhances the co-stimulatory signal and it prolongs the retention of the immunogen. That is why other than this adjuvant also increase the effective size of the immunogen. So due to these effects, that is co-stimulatory signal, increase of the co-stimulatory signal, increase of the retention time, increase of the size of the immunogen, the local inflammation increased, and as a whole the immune response is boosted. Okay. Then what is disadvantage of adjuvant? This adjuvant can produce undesirable and inappropriate response. Okay, so some side effects due to the increased local inflammation, the side effects will be increased, and the undesirable side effects, intolerance to the antigen, can be increased. So adjuvant is very commonly used with the vaccine, and in some cases it has been noted that after injecting the vaccine, the baby suffer from severe pyrexia, severe local inflammation. These properties are mainly due to the side effect of adjuvants used with the vaccines. So we should use the adjuvant judiciary. Okay.
Now the epitope. This epitopes or antigenic determinant is also very important short note. So what are epitopes or antigenic determinants? These are immunologically active region of an immunogen that binds to antigen specific membrane receptors on lymphocytes or secreted antibodies. So these are the specific site which is present in the antigen or immunogen by which they can attach with the membrane receptor on lymphocyte or the secreted antibodies. That means the it has got the complementary structure of the receptor which may be present on the membrane of the lymphocytes or the secreted antibody by which they can attach with it. So that particular region which may be composed of only 4 to 5 amino acids or monosaccharides. The whole antigen may be very large, very big, but the specific region which composed of mostly 4 to 5 amino acids or monosaccharides through which they can attach with its complementary or its specific membrane receptors which are present on lymphocytes or can attach with the secreted antibodies. This particular region is called epitopes or antigenic determinant. Their conformation may be linear or non-linear in nature. So write down this and also give some picture if the short note comes. Okay.
Next one, after going through this, we will discuss that is the uses of the antigen. So this antigen can be used to diagnose the infectious disease because the exogenous antigen present in the comes within our body from pollen, from bacteria, from virus. So they can be used to diagnose the infectious disease as well as non-infectious disease like autoimmune disease, because here our own antigen treated as non-self or foreign. So antigens can be used to diagnose the infectious disease, diagnose the non-infectious disease like autoimmune disease. It also helps us study the epidemiology of the infectious disease, and we can use these type antigen in the vivid knowledge of antigen is very necessary for the proper vaccine preparation. Okay. How we can detect the antigen? So antigen, we can use the antigen to detect the infectious disease, non-infectious disease. So then how we can detect the antigen itself? So there are several detection methods for the antigen. Most commonly used method is ELISA, that is enzyme linked immunosorbent assay, followed by immunofluorescence assay, agglutination test, immunodiffusion test, complement fixation test, etc. But most common is ELISA. Okay.
So after completing all this, we can conclude with some take-home messages. That is, first one is what is the definition of antigen? That is any substance which is introduced into the body and able to produce the antibody, that is antibody generation, are called antigen. They are mainly protein or polysaccharides in nature. They are mainly of 3 types. Few terms which you have to remember, that is hapten, superantigen, allogenic, xenogenic, autologous, etc. There are various factors which determine the antigenicity of an antigen. The epitopes are complementary sites present in an antigen for binding with its receptor in the membrane of T cell or with the secreted antibody. So they are called antigenic determinant. There are various uses of antigen and they can be detected by various methodology, most common and popular being the ELISA. So with this we complete the topic concept of antigen.
And stay home, stay safe. Thank you. Okay.